There are actually a series of abstracts at this meeting reporting on updates from the response trial with ruxolitinib. So several aspects of that response trial data are reported. The first is, and most importantly, data that was also presented at ASCO, which was the 80-week data, so about 18 months plus worth of data for patients on this study. Really important to know how these patients are doing, because we know once you start taking this drug, if you're doing well, we expect you to continue taking it. Most patients maintaining their response, no new side effects, strong, solid data. In addition, we've been digging below the surface a bit more for the data with this trial, looking, for example, at how good was the hematocrit control. Once patients had control of their hematocrit and didn't need a venesection, how long did that control last? And the data is really very impressive. Secondly, we also looked at what happened about iron deficiency. If you have a venesection, the immediate effect is you lower the hematocrit, but the longer lasting effect is you reduce iron stores and then control the hematocrit that way, but causing therefore iron deficiency, which can have some chronic health problems or complications. In the response study, we were able to show that patients normalised their iron stores while controlling their hematocrit, which is an important facet of disease control with ruxolitinib. Lastly, there is also data with regard to teasing out a direct comparison between ruxolitinib and hydroxyurea, and also looking at how patients fared, whether they were truly resistant to hydroxyurea, i.e. their disease was escaping control with the drug, or they were experiencing side effects, and that's what prompted them to enter the response study. Patients experiencing side effects entering the study were classed as being intolerant to drug. And again, we could see that responses were pretty much equal between these two aspects, or two different groups of patients. Lastly, we also saw some indication of molecular responses. That means lowering of the JAK2 mutant allele burden for patients with polycythemia vera treated with ruxolitinib.